So we finished with the discussion of stomach functions, and we moved towards duodenum. And if you'll give me 1 second, I'll make it more visible. My lights are going to be off. If you want to dim lights, you can actually do it right there in the corner. But so it's a small tube, pretty much, fairly short. Okay, so 10 inches. The entire thing is about 10 inches long. But in terms of the digestive function, especially in terms of the absorption, it is the major part of the small intestine. Okay. We've already discussed that both liver and pancreas are connected to the duodenum. They empty their exocrine secretions into the duodenum through the hepatopancreatic ampulla. Liver, that produces bile, delivers the bile through the common bile duct, and pancreas, that produces the digestive juice, delivers it through the pancreatic duct. Bile and pancreatic juice mix together and enter the lumen of the duodenum. Does that make sense? Okay. Now, the amount of chem that enters duodenum every time pyloric sphincter opens is about 3 milliliters. We talked about this. The volume of the spoon, the teaspoon, is 5. So it's a little less than a teaspoon of chem. Now this, still, it's not a solution. It's not a true solution, like a solution of salt and water, or sugar and water. It's still very, very, very small particles, maybe aggregates of proteins and carbohydrates, and fluid that contains the fragments of proteins, carbohydrates, or lipids. So this stuff has to be first digested further, and the products of nutrient digestion have to be absorbed. Okay? So they have to go from the lumen through the wall, essentially. Well, not all the way through the wall, but at least they have to reach the blood vessels. Okay? They have to reach the blood vessels. So they have to, how do we call the process when chemicals go from place where there's a lot of them to the place where there's not a lot of them? Diffusion. So these chemicals have to diffuse. Make sense? Now if we would, let's name a rate of diffusion, how fast it goes. D. Okay? Can you list me the factors, things that will increase or decrease the rate of diffusion? Let's say increase. What will increase the rate of diffusion? How can you speed up the diffusion? Okay, make the gradient steeper, right? You really cannot do that in this case. It's a great idea, so increase gradient. You really cannot do this because you kind of limit it in terms of amount of stuff that you put in. Now, consider this. If you concerned about the concentration of chemicals here, because this is where diffusion is coming from. Doesn't matter what's going on here, right? How much food you have in this area. It's all immediate gradient. But gradient, that's good. What else? What is the, yes? Size of Size of particles. Yes. The enzymes break it down to the smallest possible pieces. What is the one single most important factor that... Okay. So, fat solubility, lipid, water solubility. Yes and no, because for water soluble, it will be the number of transporters that will facilitate the diffusion. But which factor is one single most important thing that determines the rate of diffusion? Surface area. Surface area, right? So you got to increase it. The more surface you have, the faster the diffusion is going to go. Does that make sense to you? So we have to keep it in mind. I always tell when we just start AP, if you see surface modification, if you see bent surface, immediately think about increased surface area, immediately think about transport, okay, especially diffusion. Does that make sense? So, surface area of the duodenum is going to have a lot of surface modification to increase the available area for those 3 milliliters of chem to get absorbed. Okay? So there are three major modifications. First are circular folds. You can see them here. Okay? The circular folds are arranged in the spiral manner, and they're noticeable with the naked eye. They function, first of all, to increase the surface area, and second, to force the chem to move in the spiral manner. Chem does not just, does not enter the duodenum and stay there. Okay? It moves. And it moves from duodenum towards jejunum, towards ileum. Okay, 3 parts of the small intestine. That's too many L's. Yeah, I think so. Okay? And it's intestine. Okay? That makes sense? So if chem will move slower, it will have more time for absorption. Because that's another thing. If you have more time for absorption, you have better chances of chemicals to get absorbed. It takes time. It's not an immediate process. Does that make sense? So circular folds, they first increase the surface area. Second, force the chem to move in a certain way, so there is more time for absorbing nutrients. Second is villi. Now this is something I can show you here in this model, which has a freaky colors. So each of these finger-like projections, okay, is villi. If you would open up the, I need to ask my wife to send me pictures of the mouse intestines. Even when you look at the microscopic prep, you can see that they kind of velvety. The surface of the intestine is velvety because of the villi. You can see that, yes. Like velvet. Like, does anyone, like fleece? Well, something like really, really thick fleece will probably be velvety on the inside. Okay? It appears, it doesn't feel like it, but it appears velvety. So those little villi, you can see them with the naked eye. Okay? And they are macrostructures, okay, projections of the mucosal layer. So if you would look here, you'll see that white stuff here is a submucosa. Sorry, the pen is kind of off center. So that white thing, okay, that white stuff is lamina propria. That's part of mucosal layer, that connective areolar connective tissue. Okay. Now, if you look at the villi, either on this picture or here, you will see that each villus, villi is plural, villus is singular. Each villus is covered with a layer of simple columnar epithelium. Okay? Does that make sense? So we have two surface modifications now. Circular folds and villi. Okay? They become smaller as we move from duodenum through jejunum to the ileum. What do you think it suggests? Villi become smaller and kind of less frequent. Less absorption, yes. So the most absorption happens in the duodenum. And then the absorptive processes go down as we move to jejunum to ileum. Right? Now, inside of the villus, each villus, you will find capillaries. You can see the red and blue ones, okay? And the green lymphatic vessels. Now, based on your previous knowledge, what is the main function of lymphatic system? Don't hesitate. Immune. In this case, it's not immune. In this case, it absorbs fat. Okay? So, specifically, fat goes not in the blood directly, but it goes first into the lymph. That's depicted here as the green vessels. These green vessels are called lacteal. Okay? Does that make sense? So all other stuff goes into the blood, but fats go into the lymph, into the lacteal. Right? Another important structure that I want to point your attention to are those small little thingies. Okay? So-called follicles. Okay? Don't pay attention to this one, but pay attention to this one. So this is the lymphoid follicle. Now this guy is about immune response. This is so-called mucosa-associated lymphoid tissue malt. It has nothing to do with beer. Now, have you heard about Crohn's disease? Can anyone explain or tell me what it is? Mostly small intestine, yes. Well, large intestine can be involved too. Yes, it's autoimmune disease in which, in Crohn's disease, or speaking wider, inflammatory bowel syndrome, IBS, your own immune cells start to attack the intestinal tissue and destroy it. It may be fairly mild, well, quote unquote mild, leading to malabsorption, growth problems, constant diarrhea, to extremely severe when ulcerative colitis, the inflammation of the intestine that results in pretty much penetration of the intestinal wall, can lead to spillage of intestinal content into the abdominal cavity. This is called peritonitis, and patients with severe forms of inflammatory bowel syndrome often have to have huge parts of the intestine removed, and probably one of the worst part is when they have the sigmoid colon and rectum removed, and they have to have the colostomy. It's pretty much the colon is being, how to say, it opens up on the wall of abdominal cavity. Okay, so it's now why I tell you all this. It's autoimmune disease, and it is known that somehow cells, immune cells in the mucosa-associated lymphoid tissue are related to inflammatory bowel syndrome. It looks like cells that are hyperreactive, that attack its own tissue, for some reason they are directed specifically in these follicles and start to attack the tissue. So we still have no good treatment for the disease. I mean, we have plenty of treatments, but they all pretty much treat inflammation rather than underlying cause, because we still have no idea what is working there. Okay, we can chat about it a little, but it's more of a microbiology thing than ANP. Right, so we have circular folds and villi. Now, microvilli. Now imagine one columnar cell of the epithelium. Its surface, the apical surface, okay? Remember what apical means? Apical versus basolateral. Apical, that not just up, it faces the lumen, not other cells. Okay, so if you think about it, these lateral sides, they face other cells. This basal side faces basement membrane. But this apical side faces the open, the lumen. Columnar cells on the apical side, they have microvilli. I cannot show it here. I mean, I just don't have a scale. But imagine each cell has those projections. Okay? Now, those are folds of the membrane. They're not just like protein structures. Actually, membrane is like it's folded. But it's not folded in like ridge type way. It's like fingers. Imagine that there was smooth surfaces, somebody started to poke things up, poke fingers up. And you have like cone-shaped sort of. You have, of course, millions and probably billions of those microvilli on the surface of the intestine. Okay? Now, this layer of microvilli on the surface is called brush border. So that brush border actually houses enzymes. There are enzymes that are sitting there like soldiers preparing the ambush. Okay? They're not going anywhere. They are in the brush border. Okay? And they are responsible for digestion of proteins and carbohydrates in the intestine. We'll get to the enzymes. We're going to take it slow. Okay, so far we've got circular folds, increased surface area, slow down the Kim, villi, increasing surface area, and microvilli, which are very, very small, little, tiny, microscopic projections, which we will be able to see under the microscope. And in that, it's like in that brush, okay, there are going to be enzymes sitting in the membrane, breaking down the proteins and carbohydrates. Okay. Now the surface of intestine is even less. It is not that even. So space in between the villi and those crevices. Those crevices are called intestinal. Question? Yes, go ahead.
I'm having a hard time picturing the circular folds, like just putting this all together, like image-wise. Are the villi on the circular fold?
Yes.
So it's fold. Then, break it down.
Look, these are circular folds. See them? Is that clear? You see these little tiny dudes here? That budding? That's each and every of those little tiny projections is a villus. So all of them are villi. So you see they cover the entire surface of the intestine. See that? Is that clear? Did I answer the question? Good. And think about this. And each villus is covered with columnar epithelium. And on the surface of that columnar epithelium are microvilli. Now, before we get to the intestinal glands and crypts, whatever, I'm going to give you a kind of a general idea what's the difference in terms of the surface in duodenum. So imagine you have duodenum, 10 inches long, 1.5 inch in diameter. Imagine this is the rubber hose. Inner surface of such rubber hose would be, I don't remember, something like a 1 square foot or like 2 square feet. Not a lot. I mean, it's appreciable surface. Probably less than a square foot. I think it's 9 and a change inches, the page. So add some more. I mean, but still, not a lot. Now, when you throw in circular folds, villi and microvilli, we talk about 15 to 20 square yards. Now, you have 3 milliliters of Kim, and you spread this 3 milliliters of Kim over 15 square yards. It almost disappears, right? It's so thin, it's microscopically thin, and that will facilitate really, really fast absorption. Does that make sense? So you see how this increased surface area increasing the digestive activities and absorption. Right? Intestinal crypts or intestinal glands. So you have a crypt right here. When we were talking about stomach, we mentioned gastric glands or gastric pits. Essentially, this is the same. You can consider each intestinal crypt here an exocrine gland. And you have a number of cells that you can find there. Essentially, those are the cells, different subtypes of columnar epithelial cells that line the surface of intestine. Absorptive cells, kind of self-explanatory, aren't they? They absorb. That's about it. They have everything to absorb. Goblet cells. Yes, mucus production. I just remember them because of the Harry Potter. Endocrine cells. They produce hormones. So, look, it's almost the same set of cells that we saw in the stomach. Well, the difference, of course, first of all, in the stomach we didn't see absorptive cells. Right? There's not much absorption in the stomach. But here, you have absorption, you have secretion of mucus, and you have hormones. And two major hormones, the key hormones that I will write you about, I will ask you many, many times about how they work, are secretin and cholecystokinin, which has a short acronym of CCK. Right? Paneth cells, which are also cells of simple columnar epithelium, serve as the very first barrier against pathogens. They produce nonspecific antibacterial protein lysozyme, and they actually can phagocytose, so they can consume pathogens. Because if you think about this, your food is covered with a bunch of microbes. unless you autoclave it. It is covered with a bunch of microbes. Okay? Does that make sense? Now, there's one specific gland that is located, called Brunner's gland, that's located in the proximal duodenum. Now, what does that mean? What it means proximal? Any ideas? Huh? Yes. So, what's the end point? No, you're right. Farther from the end. So, if it's farther from the end point, what's the end point? No. The end point is the anus. So the part of duodenum that is farther from it, the proximal duodenum, it's closer to stomach. Okay? So this, this is going to be a proximal duodenum. So Brunner's gland that's located in the proximal duodenum produces thick mucus with alkaline pH. Why? Yeah, to counteract the acidity. Because acidic heme enters proximal duodenum, right? And in order to prevent damaging effects of the acid on the duodenal columnar cells, that thick alkaline mucus protects it. Does that make sense? Now, speaking of the replacement of the cells, I think it's about every week the epithelium is replaced. And that actually, when people are exposed to heavy doses of radiation, which kills a lot of different cells, including some of the stem cells, one of the big problems that they experience, I mean, I'm not saying that it's the only big problems that they experience. One of the complications of radiation disease is that the epithelium that is slowing off in the stomach or in the intestine isn't properly replaced because stem cells are dead. It kind of sucks. Eventually, you know, they either die or they bounce back. Now, I mentioned that intestine is the most important organ for, to cover it up, the most important organ for digestion. One single most important organ for digestion. People can live with it without the stomach. Okay. Now, there are a lot of chemical digestion processes that happen in the intestine. And before we move on with the function of the intestinal enzymes, I want to review with you what happens to the nutrients, okay? How they get digested, essentially. All right? So for this, I'm going to draw. It's not in the slides. I'm going to temporarily insert the slide, okay, right here. Is that the good? Yeah, it's a good place. I need just white page, okay. Now, what are three major nutrients that provide us energy? We're going to talk about lipids, proteins, and carbohydrates. So let's put them here. Oh, come on, man. Lipids, proteins, carbohydrates. Okay? Now, let's start with an easy one, the carbohydrates. Carbohydrates. Can you give me examples of carbohydrates that you actually eat? Bread. What is the major carbohydrate component in the bread? How we call it? Huh? No, starch. Great, starch. So we can eat starch. Okay? starch, is that a polysaccharide or a monosaccharide? Huh? Poly. Yes, great. Polysaccharide. Okay, polysaccharide. What does that mean? Poly. What does poly mean? Many. Huh? Group of. Many. Yes. Group of sugars. A lot of sugars together. Unfortunately, I have only 4. Ah! Okay. So imagine this is the. Now, I'd better draw it. So I have a group of sugars, okay, bound together. When you break it in pieces, what can you get as the result? Come on. Huh? More. Can you get more? Absolutely. What else? Yeah, disaccharides, trisaccharides. That makes sense? So you can get molecules like this or like this. Does that make sense to you? Okay, bless you. What breaks down sugars? Which chemical is responsible for breaking down starch into... That's the enzyme. Good. Amylase, yes. But you have to remember, though, that enzymes... When I screw I put the screw into the wood, okay? Do I do it with my bare hands? Well, if I'm not Schwarzenegger, then no. What I use? Screwdriver. So, enzymes, they don't directly do things. They facilitate other stuff to do things. So what is this? One of the simplest chemicals that breaks down water. Yeah. So this is hydrolysis. Okay. You add water to polysaccharide, you do monosaccharides. Does that make sense? That's what you might call it, yeast do. When you make beer or wine or moonshine, they break down and then they use monosaccharides as the food source, okay? So we figured more or less about carbs, okay? Let's talk about proteins. What they consist of? Amino acids, there you go. So you have many, many amino acids. Again, I'm going to picture it like this. Okay? But you can break them into individual amino acids. Can you have like dipeptides, yeah, you can have like this things. That makes sense? What is the molecule that breaks them in pieces? Water. Again, hydrolysis. Okay? That makes sense? The final one, lipids. So, which types of lipids you may know? Have you heard about triglycerides? Okay. That's the main source, like nutrient. The name triglyceride suggests there's something as a part of the lipid molecule. It's a molecule of glycerol. So that's the glycerol. It's actually an alcohol. And when the fat molecule is formed, instead of hydroxyl groups, you have, I simplify it a lot. So these are oxygens, okay? And these are fatty acids. Does that make sense? So each molecule of triglyceride consists of the glycerol residue and 3 separate fatty acids. It can be the same, it can be different. It's not the topic. Do you have triglycerides at home? Where? Well, what they are. What is triglyceride? That oil, yeah. Butter, that's triglyceride. Vegetable shortening, margarine, it's all triglycerides. Does that make sense? So far, for our purposes, that's enough. Now, when we break this thing in pieces, what are going to be the pieces? What are the pieces? When you break triglycerides apart. Glycerol, okay. And fatty acids. Actually, when they are separated from glycerol, they are often referred to as free fatty acids, okay, FFAs. Does that make sense? Because when we're going to talk about functions of the enzymes, I'm going to refer to these parts, okay? Amino acids, okay, peptides, monosaccharides, okay? Sorry. So going back, the brush border enzymes. So I have three major types of brush border enzymes. They sit in the membrane on the microvilli. Does that make sense? And when molecules that are their substrate appear in the brush border, these enzymes facilitate their destruction. Okay? Now, I want to remind you that when we talk about any reaction that involves enzyme, we start with a substrate. Okay? Substrate is converted into the product, and this process is facilitated by the enzyme. So, what do we mean when we say facilitated? Say, the enzyme called dextrinase breaks down dextrins into the glucose. Dextrins are fragments of starch breakdown. So if you take a molecule of starch, break it in smaller fragments, those fragments will be collectively called dextrins. It's not it's not just one single molecule, like this is dextrin. Dextrin can have 6 sugar residues, 10, 7, 8. Okay, it's all going to be dextrins. Does that make sense? Now, when we say that dextrinase facilitates this process, any idea? It means that if we mix starch and water, in maybe even foreseeable future, we talk about years probably, starch will get hydrolyzed. Years. Okay? When we add enzyme, it happens in milliseconds. So it makes it tens of orders of magnitude faster. Does that make sense? And look, our body cannot afford to function at the rates of, like, on geological scale. In 2 years, I'm going to break down this molecule. We just can't do that. Okay, we need it like this. We need it fast. So there are 4 enzymes that break down short chains of carbohydrates. Dextrinase breaks down dextrins and producing glucose. Maltase, well, breaks down sugars from malt, like malt beverages, okay? Breaks down actually maltose, which is, I think, disaccharide that consists of 2 glucose molecules, okay? Here, let me give you this example. So if this is maltose, then maltase facilitates this. and you have 2 glucose molecules. Does that make sense? Sucrase breaks down sugar, sucrose, the same sugar that we have in a sugar can. Now, sucrose consists of the glucose and fructose. And finally, lactase breaks down lactose, which consists of glucose and galactose. So what do we end up with if I had a bread with a little bit of butter on it, and well, not butter, just bread, poured some sugar on top of it, and I drank a little bit of milk and polished it all with a pint of beer. It sounds absolutely awful, I know, but I've got all 4 different types down in my intestine. So I end up with 3 different monosaccharides, 3 different individual sugar molecules: glucose, fructose, galactose. Does that make sense? The brush border enzymes make sure that everything is broken down to monosaccharides, because this is what we absorb from intestine. Not disaccharides, not long chains, either glucose or fructose or galactose. Yes?
As far as like people who are lactose intolerant or lactose sensitive, does that have to do with how much like lactase is produced
Yes.
in the body? Okay.
Yes. Inability to digest lactose, yes. And it's genetic because actually, if you never was lactose intolerant, and then when you turned whatever age you turned, you stopped drinking milk. A lot of people do that. And if you don't drink milk for a considerable amount of time, your lactose levels that are produced in the brush border will go down. Because lactose is somewhat unique, you know, because it is only in a dairy product. But when you talk about a dairy like buttermilk or sour cream, it's digested to the great extent. So if you don't drink milk but continue to eat yogurt or buttermilk, the actual level of lactose that you ingest is low because microbes did that job for you. Does that make sense? Now, if you had 20 years of not drinking milk and you will start to drink it again, get ready. And not you, well, you also, so have restroom next to you, because there's going to be diarrhea and bloating. But if you persist and you continue drinking milk, the levels of lactase will go up as the normal response, okay, to the environment. Does that make sense? So those 4 produce sugars. By the way, if anyone remembers the basic metabolism, the oxidative respiration or fermentation, doesn't matter. What is the molecule that our cells start the metabolic pathways with? One and only. Glucose. Yeah, not just sugar. It's a glucose. It's a glucose. So what about fructose and galactose? Which organ, what are we going to do with them? What do we have to do with them? Think magic. Huh? Yeah, we have to convert them into glucose. Which organ is going to do that? Huh? Liver. Yeah, it's going to be liver. Galactose is fine, but fructose messes it up. Okay? So these 4 carbohydrate. Okay? I'm going to skip enterokinase for now. I'm going to talk about it, the last one. The next 2 are aminopeptidases and dipeptidases. So aminopeptidase is the enzyme if you have a chain of amino acids like this, each chain like that has amino end. Wait a minute, let me start it over so you can see it better. Say you have a chain of amino acids. Okay? One end is going to be amino end, and another one is called, my handwriting is awful. Another one is carboxyl end. Okay? That's amino, and that's carboxyl. Aminopeptidase chews off one by one amino acids from amino end. Does that make sense? Imagine a train that goes, down the railway, and there is a monster that's chasing this train and biting off car by car, one by one. Does that make sense? Specifically from the back part, not from the locomotive side, okay, but from the back side. Does that make sense? Okay? Dipeptidase is even simpler. Dipeptidase takes the dipeptide and breaks it into two individual amino acids. Okay? Does that make sense? Finally, nucleosidases and phosphatases, they take a nucleotide. And I want to remind you that nucleotide, sorry about that, nucleotide is the molecule that consists of ribose, sugar, nitrogenous base, let's call it a B, and the phosphate residue. I'm not going into chemical details for ribose and stuff like that, okay? Now, nucleosidase and phosphatases, they break it into individual phosphate, individual nitrogenous base, so phosphate, nitrogenous base, and ribose molecule. Okay, so they break down each individual nucleotide in pieces. Before we talk about enterokinase, look, like kind of take a virtual step back and look at this enzymes. Look at the enzymes that digest carbs, enzymes that digest peptides, and these ones. Do you see anything in common? Well, that's because the enzymes. Huh? Okay. Hunter first, you second. The name is specific, so you can tell by the name what the substrate is. What? Even bigger picture. The molecules that they break down, are they really, really large like the proteins, or are they rather small? Okay, sucrose. Yeah. Small. Galactose. Oh, lactose. Small. Now, look at the protein stuff. Dipeptide. Small. It's not the entire protein, it's just a short peptide. Nucleosidase and phosphatases, they aren't dealing with the entire DNA molecule. They deal with individual building blocks of it. You see? Now, think about this. Those dipeptides, tripeptides, dextrin, sucrose, lactose, they are by themselves maybe products of the breakdown of even larger molecules. Does that make sense? Imagine that you have a brick wall, and you want to destroy it in pieces, just complete pieces, just brick by brick. Okay? And you have a sledgehammer. And when you hit the wall, the first thing that's going to come out of it is the chunks of bricks. And then each chunk you can break in smaller pieces. Does that make sense? So those substrates are chunks that brush border enzymes break in the smallest individual pieces. They finally digest peptides, products of the protein degradation, into the amino acids. They digest smaller, what we call oligo means few, saccharides into the individual monosaccharides. They digest nucleotides into the elementary building blocks, such as phosphate, various nitrogenous bases, or ribose sugar. Does that make sense? That idea of breaking down chunks of the bigger molecules. And that happens specifically in a brush border. Okay? Now, before we move on to enterokinase, what would happen next to those small things like glucose and amino acids? What should happen to them once brush border enzymes broke down those chunks, so you have those individual elementary things? So how do we call this? Well, diffusion mediated. So they are absorbed, right? Taken, absorbed. Okay. So the next thing for them would be to get absorbed. Does that make sense? Enigmatic enterokinase. So this is an interesting enzyme. Now, if you think about the processes that happen in the brush border, we just figured out that brush border enzymes deal with the smaller chunks of larger molecules. But then you have to ask yourself, those smaller chunks, what makes them? Okay, where are those enzymes that break down huge molecules like a big protein or the starch molecule? Okay, where are they? Turns out those enzymes are in the lumen of intestine. Does that make sense? So they are in a slightly different location. Okay? Again, when I say in the lumen, it doesn't mean that you have a tube like that and there is a proud enzyme floating right in the center of it. Lumen's empty. Seriously, lumen itself is empty. There's nothing in there. Because everything is on the walls. It depends how close it is to the brush border. So enzymes that break down starch, proteins, they are farther from the brush border. Okay, kind of on the outskirts of that layer of chyme that covers the walls of intestine. There's a ton of enzymes, and the function of enterokinase is to activate the enzyme called trypsin. Enterokinase converts the molecule of trypsinogen into the molecule of trypsin, and therefore activates the entire digestive process of the big molecules. So it's kind of interesting that brush border breaks down the products of other enzymes, but in turn activates those other enzymes. Does that make sense? We're going to chat about secretin and cholecystokinin, and then we're going to take a break. And we're going to look at some micro slides, and we're going to take a quiz at the end of the class.
I have a question about, you said like you would live without the stomach.
Yeah.
Where, so like if it was just your esophagus and then your duodenum, how like you would miss out on all like a lot of that mechanical and stuff. So would you just have really poor like absorption?
Of course. I mean, your digestion would not be perfect. I'm not saying that you're gonna have perfect, wonderful life without the stomach. I always give example which organs are not critical for life. So if you think about this, so what's not critical for life? Stomach. Usually they leave just a little piece of it. Okay. What else can you think of? Large intestine. You can get this out. One lung. The entire gallbladder. One of your kidneys. You can technically get rid of the urinary bladder, but you got to have some sort of a bag right there. Well, I'm not talking about eyes, ears. It's kind of depressing. I think, well, spleen, you can get rid of the spleen. I mean, and you can still live without those organs. I'm not saying that you're going to enjoy your life, but you're gonna survive. Okay? So when there are some less and more kind of damaging, you know, and we will see that removal of large intestine doesn't affect your quality of life that much. Yeah. My grandmother was diagnosed with a colon cancer, got like 3 feet of large intestine removed. She was 73 at the moment. She died when she was 82 of completely unrelated reasons. Just totally. She got Alzheimer's later. But it was just, you know. So we can, small intestine, different story. The malabsorption, there's going to be a lot of troubles. Okay. Same goes like spleen. It's fairly okay to live without the spleen, but living with one lung, it kind of sucks. Although you can. Okay. So let's talk about hormones. Now think about what's going on in the duodenum when Kim enters there. 2 types of stimuli. One is mechanical, distension. Does that make sense? Second is chemical, right? There are 2 major chemical stimuli. One is acid. It comes from the stomach. Another is fat. Also comes from the stomach. Does that make sense? Just this statement, that from the stomach, acid and fats enter the duodenum. Do you agree with me? Does everybody get this idea? Now, the entire system works by negative feedback. So if you get acid somewhere, what do you have to do with it? Huh? No. What I'm saying is, you have a part of the digestive system, the duodenum, which is not supposed to be acidic. And suddenly, you have acid in there. What you got to do with that acid? You have to neutralize it. How do you neutralize acid in the kitchen? Well, do you have... Yeah, you have a can that says base. No, water is neutral, well, slightly acidic. So what is the base in the kitchen? Baking soda. So you got to take baking soda, well, the volcano experiment, okay? You got to take baking soda and put it onto your acid. Exactly. Pancreas does exactly this. Pancreas produces bicarbonate, okay, which neutralizes the acid. Does that make sense? That idea. So that's your negative feedback. Acid, bicarbonate, neutralization of acid. Fats. What you can do with fats? Well, before storing it, you have to, it's a nutrient, you have to digest it, yes. So, you got to release enzymes, right? Pancreas produces enzymes to break down fat, right? Fats are going to get broken down, absorbed, and you find. So that cheme that accumulates in the intestine stimulates activities of pancreas, and as we will see in a second, activity of the liver that facilitate the breakdown of nutrients and their absorption. Okay? So, let's take a look at this flow chart. So first of all, This horrible green fluid is chyme. Okay? The red dots are CCK. The yellow dots are secretin. Those are two hormones that are produced by the enteroendocrine cells in the small intestine. So when chyme enters the duodenum, it causes the release of cholecystokinin and secretin into the bloodstream. They are true hormones. Does that make sense? Now, the exact stimuli for the release of those two hormones are acid and fat. Does that make sense? CCK, the red dots, stimulates production of digestive enzymes from the pancreas. Right? That makes sense? Secretin also stimulates pancreas to release bicarbonate to neutralize the acid. So to some extent, you can say that acid stimulates secretin. Fat stimulates CCK. Because these hormones mediate the response that gets rid of the stimulus. Does that make sense? Secretin induces release of bicarbonate, okay, that destroys acid, neutralizes acid, and CCK stimulates the release of pancreatic enzymes that break down fat. Cool? Not only this. These two fellas travel farther. Well, not farther. Can't really say because they're systemic. They stimulate the liver. So secretin, both of them, CCK and secretin, okay, stimulate the liver to release bile, to start producing bile. Okay? And if it's between the meals, we definitely have a little bit of a bile stored in the gallbladder. So CCK stimulates gallbladder to contract and squeeze the bile out into the duodenum. Right? That makes sense? Now, that bile is what you need to absorb lipids, digest and absorb lipids. Does that make sense? So CCK is produced essentially in response to lipids or fats, whatever you want to call them. Does that make sense? So you have pretty much one type of response to the acid, which is bicarbonate production by the pancreas, and two types of response to the fats. Enzymes from the pancreas and bile from the system here. System that consists of liver and gallbladder together, okay? Gallbladder is the kind of first responder. Just stores the bile, stores and concentrates it. Gallbladder does not make the bile, okay? Just dumps it on the fat, and then liver kind of gets in the loop and starts to produce more bile. Does that make sense? Okay. Mechanical digestion is extremely simple. So first of all, there's a peristalsis, okay, which we talked about so many times. Just slowly moving the chyme down through the small intestine. And, important, segmentation. Because think about this, you have chyme that just entered the duodenum from the stomach, and you have digestive enzymes and bile and all those juices. You got to mix them. So what happens, circular muscles, smooth muscles of the duodenal wall, they contract in different places. Okay, essentially mixing it up. Does that make sense? That's the mechanical digestion. So you have about, food have about 5 hours to get digested and absorbed, partially absorbed in the duodenum. Okay. Okay.
It doesn't take that long for like the one release. It's like the chem keeps getting released from the stomach into the blood. Right?
Of course, it doesn't mean that those 3 milliliters stay there for 5 long hours. No, it means that, say you have first portion of chem that entered the duodenum. It will leave the duodenum, the same, well, quote unquote, the same portion, the same, those 3 milliliters will reach the end of duodenum in 3 to 5 hours. Okay, but it's a continuous process. You have to, yeah, it's never, okay, we've got the whole bunch, let's throw it in the duodenum. We've dealt with the entire, I don't know, T-bone steak, yeah, and then just dump it into the large intestine. Okay, it's a continuous process. Does that make sense? Let's take a break.